Properties of biological macromolecules. So to start off, the main molecules of life are carbohydrates, lipids, and proteins, as well as nucleic acids. What are macromolecules, you might ask? Well, some of those molecules are pretty big, so there are many smaller parts that make them up. The three main structures that have this are going to be carbohydrates, proteins, and nucleic acids. Lipids aren't really going to be included in this, and we'll talk about why in just a second. So carbohydrates. Their main function is going to be to provide energy and structure, and this is going to be in the form of a more short-term kind, as they get used up very, very easily. Something you may want to keep track of is that shape and placement are extremely important to functioning in biology. In carbohydrates, this is seen as a placement of the hydroxyl, which is going to be right here. The monomer of a carbohydrate is called a saccharide. This could be in the form of a mono, di, or poly, meaning one, two, or many. The most common type you'll see is a glucose, which is going to be C6H12O6. This is a building block of life, meaning it's in almost every single form of life. It's always found in the ratio 1 to 1, and as a diagram, you'll see it as a ring and a more standard one, as seen right here. The ring is just that it's going to be present with water as well. The bond name is called a glycosidic linkage, basically meaning that it can be straight across like this, or it can be a more lattice-like structure. The lattice is mostly seen in plants and is the reason why they're able to have such strong structure and support themselves. The reaction that actually brings these together is called dehydration synthesis, basically meaning that water is produced from the reaction. We'll take a closer look right here. So basically what happens is the OH and the H come together to form water, leaving behind an oxygen. This oxygen will basically bring the two saccharides together, forming a little chain. The opposite of this is called a hydrolysis reaction, basically meaning that water is instead added to break the bond. Next up we have lipids. Their main function is to provide energy in a more long-term way, kind of as needed. Their bonding type is called an ester linkage, and they actually don't have any official monomer. This is because they're not big enough to have it. The first type is called a triglyceride. It has a glycerol head and a fatty acid tail. The two types are called saturated and unsaturated. Saturated comes in a more straightforward kind of thing. It's completely straight and is completely covered by hydrogens. Because of this straight kind of format, it stacks very, very easily, so at room temperature it tends to be solid like butter. Unsaturated tends to be bent, and this is because of double bonds instead of single bonds like in the straight kind for saturated. Because of this, it doesn't stack very well, so at room temperature it tends to be very liquid, like olive oil. This can be reversed and is seen as margarine when hydrogen is bubbled in. The second type is called a phospholipid. They're made up of a phosphate head and two fatty acid tails instead of just one. They form the outer lining of cells and regulate everything that goes in and out. This is seen as a phospholipid bilayer, like this. Basically, the phosphate head, has a negative charge and is hydrophilic, meaning that it really likes water, but the tail, which is not charged at all, is hydrophobic, meaning it does not want to be around water. So when it forms this phospholipid bilayer, it actually forms so that the tails are on the inside, protected from the water, and the heads are on the outside, still in contact with the extracellular and intracellular fluid. The last type is called cholesterol. Basically what this does is that it maintains the fluidity of the cell membrane. It does this by grabbing onto phospholipids, so when temperature gets too high, it holds them together, and when temperature gets too low, it actually separates them, just like that. So next up we have proteins. So what is a protein? A protein is a biologically functional molecule made up of one or more polypeptides, each folded and coiled into a specific 3D structure. Their monomer is a amino acid, and their bond type is a peptide bond. The most universal thing about this is that they all have a backbone, which is made up of a amino group, right here, an alpha carbon, which is going to be just here, and a carboxyl group, which will be right here. The thing that actually separates them from each other is their R group, which just kind of tacks on at the end, which you can see highlighted in green, and is kind of the thing that makes it have its chemical properties, and also gives it its 3D structure. So what is the difference between a polypeptide and a protein? So a polypeptide and a protein, they all have the same general makeup. But the thing that makes the protein is the polypeptide going through the folding and coiling process. And this is normally a 4-step process called the primary, secondary, tertiary, and quaternary. I'm not going to go too much into that, but it's extremely important in creating the actual protein. Finally, we have nucleic acids. Their function is going to be to provide directions for its own replication, and although it isn't too involved in the next two, it also directs RNA synthesis and gene expression, also known as protein synthesis. Its monomer is a nucleotide, which is made up of 3 main parts, and that's going to be the 5-carbon sugar, also known as pentose, a nitrogenous base, and a phosphate group. The bond name is called a phosphodiester bond, and the two most popular things you'll probably have heard of are DNA and RNA. The difference between these is that they have different sugars. So DNA has deoxyribose and RNA has ribose. So the main difference between these sugars is that deoxyribose actually lacks an oxygen atom on the 2nd carbon in the ring, which you'll see here and here. The second most important difference between these is that DNA has a double helix, RNA has a single. DNA also has a anti-parallel kind of system, so it has a 5-3, and then the other one will have a 3-5 when they match up together. The last and most important difference between these is that DNA has different nitrogenous bases. So they'll have thymine, cytosine, guanine, and adenine. However, RNA will not have thymine. Instead, it'll have uracil. Thank you, and I hope that helped.